I believe the impact on organ-on-chip technology regarding the future years is going to be just exploding, because there are already studies together with the regulatory authorities, not only in Europe but also in the United States. And I think including these organ-on-chip models into regulatory frameworks is going to be really important for drug developmental processes. Hi, my name is Kaden. I'm a doctoral candidate at Dynamic42 and also affiliated to the University Hospital in Jena. And I mainly work as a scientist in the laboratory on cellular-based OOC models, but also on the further advancement of technical equipment and the chip periphery. I joined the company working on the liver-on-chip model, and this liver-on-chip model I use for studying drug-induced liver toxicity. So I mainly use this to evaluate adverse hepatotoxic side effect of different drug compounds. I think this is really relevant because we see it nowadays in a lot of patients that they suffer from these adverse side effects, which have not been previously demonstrated in animal models or simple cell culture models. So I think this is a really crucial topic that is going to be very important in the future. And then I further continued here at Dynamic42 with my dissertation. So I shifted a bit my topic. I'm more interested now in the intestine-on-chip model, where I simulate disease conditions by inducing inflammatory bowel disease. And I use this disease model to investigate the effectiveness of postbiotic treatment, but also I want to see changes or alterations in the host microbiota interactions. So what inspired me most to work with organ-on-chip technology? I think it was in the first place because of the high innovation potential of the chip. So when I first read about it, I was like, wow, you can really recapitulate small functional organ units inside these little plastic chips. This was very new for me, and I think also for a lot of other people when this technology came up. And when I started working with this organ-on-chip model, for example, the liver model, I realized, okay, this is not really only an innovation, but it can also in part be an alternative for animal models. Because of the animal models, you have highly animals suffering during the experimental processes, and it takes also a high financial cost, but also a load of bureaucracy. So the most exciting project I'm currently working on is based on the intestine-on-chip model. So this is what I basically do during my dissertation thesis. But we are also currently developing this intestine-on-chip a bit more by including primary intestinal epithelial cells, and then in the next step design a completely primary-based or stem cell-based intestine-on-chip model. And this model is not only primary cell-based, but will also include a lot of biochemical but also electrical sensors to really have a real-time measurement inside of this organ-on-chip model. And furthermore, also to include oxygen gradients for host microbiota integration. In terms of assay opportunities, but also experimental readouts, organ-on-chip technology is not really limited, but it does not really differ also from conventional assays you might be familiar within your own laboratory. So basically you can do all of the principal cell culture assays, for example, colorimetric assays, ELISA. You can also stain the tissue for important immunofluorescence markers and then check the morphology at the fluorescence microscope. But also investigation with the light microscope is definitely possible. Furthermore, we can also measure barrier permeability, which is really important for the intestine-on-chip. But you can also extract the tissue by cutting out the membrane and then lyse the cells for further downstream analysis like RNA isolation, DNA isolation, and then also sequencing of the RNA would be possible, for example. So my recent breakthrough was really a cooperation publication together with the Leibniz HKI, where we published about the Candida albicans infection model in the intestine-on-chip that we provide at Dynamic42. And this model is particularly very interesting because it combines also the interdisciplinary fields of microbiology, but also organ-on-chip technology. And we further combine it with advanced image analysis, which is also a really cool feature that can help in the future to really investigate the tissue in depth. One of the most important misconceptions, and I hear this a lot of times during conferences, but also customer calls and meetings where we interact with people that are not familiar with organ-on-chip technology at all, is that organ-on-chip technology can replace animals from one day to another, and we will never do animal experiments anymore. This might be a misconception because the organ-on-chip is a fit-for-purpose tool, and I think it can really help to answer scientific questions that you already have or that you want to discover. But it gives you more an investigative field of view in the preclinical phases before you go into an animal experimentation. And if you already see that, for example, your drug is having a toxic effect in your model, why would you go then a step further to an animal model? You can just leave this out. So it helps you to really prevent excessive animal use, but it's not a full replacement at this time.